Assalamualaikum and good morning. My name is Nur Faraya binti Saleh and my metric number is 201725. We are from Group 2 and today we are going to present about production of biopharmaceutical maps using mammalian cell lines. Next slide. Firstly, what is MAPS? Monoclonal antibodies are identical immunoglobulins generated from a single B cell clone. These antibodies recognize unique epitopes on a single antigen. These antibodies are produced by cell lines or clones obtained from animals that have been immunized. Next slide. Why we need these maps? Maps are important reagents used in diagnosis disease and in treatment for infections and cancer. It is also important for identification of proteins, carbohydrates, and nucleic acids. We can also control cell replication and differentiation, advance our knowledge between molecular structure and function, and can also understand more about host response to infectious disease agents and toxic produced by these agents. Next slide. Why we use mammalian cell line in producing maps? Mammalian cell types have become dominant due to their ability to produce large amounts of map with a consistent quality and to adapt well to culture in large-scale suspension bioreactors. These cells also have capability to perform the required protein folding, assembly, and post-translational modifications. So the map produced would be biochemically similar to human forms. Next slide. Next, we'll look at what kind of cell is used to produce maps and why. There are three types of cell that can be used. The first one is CHO cells, which is Chinese hamster ovary cell. CHO cells quickly gain recognition for ease of culture and fast generation times. Pathogenic human viruses do not replicate in CHO cells, therefore it will increase the safety of the map produced and simplifying the downstream purification process. These cells also easy for genetic modification and also have record to be produced safely, biocompatible and bioactive maps. Next slide. The second one is murine lymphoid cells. Murine lymphoid cells have the innate ability to produce large amounts of immunoglobulin. However, CHO-derived cells are becoming the preferred host because glycoproteins from NS0 and SP2/0 can have residues that are immunogenic and have a reduced in viral half-life. Slide. And the third one is human cells. These cells do not carry any antigenic carbohydrate groups, therefore it can use for production. The cell line can reach the cell densities therefore higher than CHO cells. However, regulatory concerns exist because these cells lack of resistance against adventitious agents. That's all from me. Now I will pass the presentation to the next presenter.
Okay, my name is Adham. So there will be two problems for the production of MAB, which is monoclonal antibody. The problem one will be they need to generate stable cell line to create the successful clone due to apoptosis that affect health and lifespan of culture that probably kill the culture and the cell. And for the problem two, there will be the screening and selection of high productive and stable clone from the transfection population in a limit time frame is very, very challenging for the producer of the monoclonal antibody. Okay, the next slide. So the first cell line development, there will be two processes that we need, which is the first is the vector construction and then the transfection process. So in the mammalian expression vector, it will consist two space or we call it two cassette. The first cassette will be antibody gene and selectable marker gene space. And the second cassette will be for the plasmid replication gene. For the first cassette, it will be for the expression of the vector itself for the production of antibody. And the second one for enable the replication to occur in the vector. And then for the vector construction, the function of the vector construction is to drive antibody heavy chain and light chain reaction, which is in this case, we need a strong promoter or enhancer that we use, such as cytomegalovirus and elongation factor, which is EF1 alpha, in order to achieve the high level antibody expression. And then the cell will be having a transfection process, which is this process will be to stabilize the introduced vector DNA in mammalian cells. And there are four methods that can be used, which is the first is calcium phosphate method, electroporation method, cationic lipid-based lipofaction method, and the last one, dendrimer-based method. But only electroporation and cationic lipid-based lipofaction that are very famous being used in the production. And the transfected cell will be categorized into the different selectable marker, which is two. The first one will be metabolic selectable marker and the antibiotic selectable marker. Okay, next slide. So The last one will be a screening process, which is usually used in the commercial and clinical, and they usually use the ELISA assay to screening the cells, and this with this we can observe the cell growth, specific cell productivity, and the volume of the monoclonal antibody itself. And then the top 12 anti-24 clone from the 1,000 of a clone, we will usually take as a candidate to be analyzed in the fat batch cell culture for the in bioreactor. And then further, the top 4 anti-6 clone will be taken as a final product, which is it can contribute to growth, specific productivity, volume, and loan stability. Also, the glycolation profile, which is this last top will be a single clone that can be chosen for the mass production of monoclonal antibody. Okay, I pass for next presenter.
Okay. Apopto- Sorry. Apoptosis is where the cell dies due to high stress condition. Delaying apoptosis can be done by overexpressing anti-apoptotic genes, which not only increases the cell viability but also increases the monoclonal antibody production. Besides, degenerating or decreasing the pro-apoptosis gene inhibits the activation of downstream caspases when apoptotic stimuli is present. Glycosylation is an important process for the production of the pharmacokinetics products. Genetic approaches are considered to be more efficient compared to cultured conditions. Monoclonal antibody is known to function well by eliciting the antibody-dependent cell cytotoxicity. Hence, the ADCC could be increased through afucosylation in where when there is no fucose sugar units present in the oligosaccharide units, which also eventually increases the sensitivity of the antibody. This method is currently considered and approved as the most successful method in this industry. That's all from me. Thank you.
The next part is about vector design for efficient expression. Next slide, please. So the first strategies is about codon optimization. So it can improve the MAB production so we can get a high yield of MAB. Codon optimization is important due to the differences in tRNA abundance in different cells and effect on the mRNA stability and the secondary structure formed by the transcript. So the earliest attempt for codon optimization is through the empirical method of DNA shuffling, which improved the expression of GFP in CHO cells. The new system called codon adaptive indices will able to score the optimization of the gene from the value of 0 to 1. So the closer the value to 1, it will indicates the high expression of the gene. It also perform comprehensive assessment of codon usage, which prevalent for choline, threonine, aspartate, and cysteine. Next slide, please. So the next strategies is co-expression of light chain and heavy chain. Efficient expression of LC and HC genes require an appropriate signal peptide for proper folding, assembly, and post-translational modification for MAB assembly. So LC-HC peptide ratio plays an important role in the kinetic of MAB formation. There are studies that reported that excess LC is beneficial for higher MAB expression level, and the ratio is if the ratio is above 1.5, it will resulted in minimal product aggregation levels. Next slide, please. So this is the vector design for LC and HC MAB production. The first one is co-transfection on two separate vectors shown in A. The second one is single vector with separate expression cassettes for each gene shown in B. And the single vector, single promoter vector with genes linked together on one expression unit shown in C. Scientists utilize most C system. Next slide, please. So the last one is the selection strategies. So there are three types of selection marker gene: resistant towards antibody, overgrowth advantage in nutrient deficient condition, DHFR, and also GS. But I will only touch about the hydrofolate reductase, DHFR. So DHFR is used together with the DHFR deficient DG44 or DXP11 CHO cells. It involves the reduction of the hydrofolate to tetrahydrofolate, which is needed for the nucleic acid metabolism. So the selection is conducted in the media that is devoid of hyperxanthine and thymidine, and then the amplification can be further carried out by adding MTX, which will inhibit the DHFR activity. So in order to survive, the cell will start to amplify the DHFR gene copy. And the MAB gene, which located on the same transfected vector or nearby the site, will also amplify, thus increase the MAB production. So there are two new selection method that is still under studies, which is amplifiable selection marker based on hypoxanthine phosphoribosyltransferase transferase and also toxin and antitoxin method. That's all from me. I will pass to Chen.
So I will continue with the third improvement in production of MAB, which is the high throughput clone selection, which means select the clone in high quantity. So scientists utilizes flow cytometry to select the cell line that are producing MAB from those that are not. And in order to increase the sensitivity of the flow cytometry, scientists actually utilizes another type of flow cytometry technique, which is known as FACS. So FACS is an abbreviation for fluorescent activated cell sorting, where it adds a degree of functionality to the flow cytometry. So by utilizing highly anti as well as to sort to differentiate the biological sample by a nearly limitless number of different parameters. So for example, FLEX is widely used in the analysis of whole human blood, as well as to sort and analyze the cell line for various biological assays. So in our case, we want to select the cell line that are able to produce monoclonal antibody. So before I move on to the process, what is flow cytometry? Flow cytometry, as we all know, is a powerful tool that allows simultaneous multi-parametric analysis of the biological sample for up to thousands of particles per second, meaning that within 1 second, there are thousands of particles can be passed through the flow cytometry and can be sorted into positive sample and negative sample. So in our case, we wanted to obtain the cell line that are able to produce MAB. So we will first label the chain with orange and green fluorescent respectively on the light chain and the green chain. So when the laser hits on the cell line that has both the fluorescence, they will produce data, and the data will be collected by the detector. So the more the antibody produced, the higher the fluorescent signal will be. And since we label our antibody with two types of fluorescent, which are orange and green, so we are expected to get 4 types of cell, which are cell ABCD. But we are only focusing on cell type C, which are the cell that can express both orange and green fluorescence. So like Azmina mentioned before, the ratio, the LC to HC ratio is crucial for the production of functional monoclonal antibody. So that's why we are targeting only the cell that are the cell line that are producing, that are expressing both orange and green fluorescence, because only when both gene on this light chain and heavy chain are expressed, we can only get the functional antibody, monoclonal antibody. Other than that, like for example, cell A, which is the cells that are only expressing green fluorescent, it will not produce functional monoclonal antibody because the gene on the light chain is not expressed. So in short, mammalian cell line is chosen to be the most widely used cell line in producing monoclonal antibody because it is the safest and it is the most effective cell line that can produce monoclonal antibody. And the monoclonal antibody produced is more similar to human. And mammalian cell lines are able to produce high amount of MAB with a constant quality, like mentioned by Farahia. And the monoclonal antibody produced by mammalian cell line are capable to have post-translational modification such as glycosylation. And to summarize, there are 3 improvements that have been done to shorten the production time, which are host cell engineering, which involves the over expression of the anti-apoptotic genes as well as downregulation of the pro-apoptotic genes. And the second one is the vector optimization, which utilizes the codon optimization, which measure the optimization of the gene for expression of gene. And the last one is the high throughput clone selection, which I mentioned before. The scientist utilizes the flow cytometry technique to sort the cell into those that can produce the cell line that can produce functional monoclonal antibody and those that are not. So as conclusion, instead of looking for ways to reduce cost through increasing the titers of the monoclonal antibody, meaning the concentration of monoclonal antibody produced, scientists now have shifted towards to shorten the developmental timeline as well as to produce improved quality of monoclonal antibody. So I think that's all from us. Thank you.